I'm Christina Pavloudi. I'm going to present you my talk on investigative microbial ecology in extreme habitats. I am a microbial ecologist, marine biologist, postdoctoral scientist, data scientist. And I'm working currently at the EMBL-EBI, the European Marine Biological Research Center. So first, a few details on what is EMBL-EBI. Some of you may already know it, but in case you don't, EMBL-EBI is a research infrastructure. It was founded in 2013, and it gained the ERICA status in 2018. It's an organization that enables researchers to use facilities, resources, and services in order to conduct groundbreaking research and foster innovation and society in general. So EMBL-EBI is composed from has members from 9 European countries, and it's comprised by 70 marine sites. One of the major projects that EMBL-EBI runs is the EMBL-EBI, or else the European Marine-Omics Biological Observation Network, which is the first coordinated genomic observatory in Europe with 17 participating marine sites. And if you want to read more about it, there's a relevant publication. So in a nutshell, EMBL-EBI advances marine research and providing cutting-edge services for scientists, academia, and industries throughout its network and its marine stations. So back to our subject on extreme habitats and extremophiles. What are extreme habitats? Extreme habitats are environments characterized by harsh conditions, so something that is beyond the optimal range for the development of most organisms. Likewise, we have the definition of extremophiles, which are organisms that are adapted to survive and thrive in those harsh environments and conditions. But just on a related note, just have in mind that the definition and concept of extreme presupposes that there should be a definition of normal. So what I'm trying to say here is that most of these definitions of extreme came from our own human perception of what is normal and what is not, where we could thrive and survive and where we couldn't. But this isn't the case for the majority of the organisms that actually live in those environments. If you would go in a hot spring, for example, they would consider microorganisms living there, they would consider everything as perfectly normal, and they would consider what we live in as the extreme. So just to have in mind, this perception. So there are some types of extreme environments that are very popular and very well known. Some examples are hot springs and hydrothermal vents, so in general, geothermal habitats. For example, this is an image of the hot springs in Yellowstone, which is probably like the one and only hot spring that people all around the world know about. It's the most popular, let's say. And hydrothermal vents are found, in the deep ocean. This is a picture of the black chimney there. Another type of extreme habitat are deserts. Because I think it goes without saying that it's a matter of rainfall and drought, which is what causes the extremeness. This is a picture of the Atacama Desert, one of the largest on Earth. Also, another type is the acid mine drainage. Here is a photo of Rio Tinto, which is considered extreme, and both from a natural but also from an anthropogenically affected way. And it's also considered as a Mars analog site. Other types of extreme habitats are permafrost and glaciers, as you can see here, but also even salt evaporation ponds. Because here, for example, high salinity would be what pushes the limit of life. So having said all these definitions from extreme environments and having seen all this type of extreme environments, we come to the types of extremophiles. So the organisms that love living in those environments. We can have all kinds. We can have extremophiles depending on pH, which are acidophiles, so those that like lower pH values, alkalophiles, those that like higher ones. We have temperature-dependent extremophiles, the thermophiles, which have high temperatures, psychrophiles that can go even below 0. Of course, we have salinity-wise halophiles. We have pressure microorganisms like, pressure, barophiles or piezophiles, as we may call them, even radiation and metal concentration, drought, as we said before. And even oxygen in some cases can be considered type of extremity, even though it's really hard to put it into perspective and into concept. The thing is that in many cases, some of these categorization occur simultaneously. So we can have a hypersaline environment where we have organisms also thriving in high temperatures. We can have combinations of those. So sometimes it's a bit difficult to identify if there's only one pressure driving the extremity or not. So why are we interested in those extremophiles? Because many of them have very effective DNA repair and replication mechanisms. They use enzyme-controlled biochemistry that is unique in a way, so it allows them to counteract all the harshness of the environment and the hostility of the environment. So they have adaptations at the membrane level that we see different composition and types of lipids, different amino acid composition and structural adaptations. And we also see that they can produce biocatalysts that are unique and highly specialized so that they can perform the same reactions that all the other normal microorganisms can perform, but in the extreme conditions that they live in. So extremophiles can produce extremozymes, which are basically a definition for their enzymes. They have a more compact structure and they have increased number of hydrophobic interactions, hydrogen bond networks, disulfide bonds, and so on. So they can help us, we can use them to design new generations of genetically engineered enzymes and have novel biotechnological applications. Applications in several fields, as in agriculture, chemistry, biomedicine, and so on and so forth. But apart from this exploitation that we can do in the extremophiles and the interest that we can have in them in that way, they're very interested in the way that they can help us know the knowledge of limits of life, because the more we investigate them, the more these limits expand. They can help us develop hypotheses on how life formed, what was the origin of life, how was the planet when that happened. And they can have implications for extraterrestrial life, because some of the conditions in extreme environments seem to be very similar to what we have found in other planets. Let me see. I'm sorry for a moment. Okay, sorry. So what are the methods and challenges that we come across when we try to study extremophiles? Well, in a nutshell, they are... Oh, sorry. Spoiler alert. Sorry. In a nutshell, they are similar to what we have and what we can use to study all other kinds of microorganisms and all other kinds of ecosystems. However, there are a few challenges, and those sometimes have to do with locations that they are difficult to access and sample. So, for example, you see me in this picture, and I'm trying to sample a hypersaline swamp, but it was really difficult to actually step on the swamp without getting swollen by the mud. So this was the solution that I found to actually, collect the sample. In other cases, locations can be difficult to access. For example, we can have caves where we need special equipment and special training. Not everyone can actually go there. And because of these cases, because it's difficult to access and sometimes it can be very expensive, study material may be limited. There isn't an endless supply of material that we can use to study, microorganisms in extreme habitats. Sometimes we are part of big sampling campaigns, and we have to, let's say, divide one sampling between many different disciplines. So then it's even more difficult for us. And because of their intrinsic environmental characteristics, sometimes it's difficult to extract optimally DNA and RNA from those samples. We may need to modify our protocols for the DNA and RNA extraction. Yeah, and then we need to work extra in that. And another challenge that we always have to take into account is that when we go in these strange places, we never know what we can discover. We never know what might come out. So we might find a highly resistant microbial strain, as for example, these colleagues in the paper I cite here did. So if we don't pay enough attention, we can spread antibiotic resistance to other microorganisms. So we need to have, to be careful about certain things. But as far as the methodology is concerned, what we normally do is that the first step that we were trying to do for years, and the first step that we try to do in many microbial ecology studies, is to culture microorganisms. But of course, culturing in extreme environments might be a bit more difficult than in, let's say, again, normal habitats. So we need to modify a bit the way we approach culturing. A way to do that is to employ omics results into into the way we can approach microbial cultivation. So one way to do that is to do a function-guided cultivation, as the authors of Young et al. Mentioned it. So what does this mean? Is that we can go from the sample collection, we go through metagenomic sequencing, binning, and then we do a functional analysis of our sample. So we try to identify what are the key functions in the sample, what is missing from it, so that we can design the perfect medium to try and isolate new microorganisms. Because if we don't design new media, then we will keep using the same ones, and we will keep isolating the same microorganisms again and again. So one way we can achieve that is through MediaDive. MediaDive is a database repository, I think we can call it both, from DSMZ, which is the German collection of microorganisms, one of the biggest in the world. So MediaDive already contains a very long list of available media with the recipes. It's like a big cookbook, but for every microorganism that we can culture. So you can search it based on the taxonomy, and you can identify the media you can use to isolate taxa of interest. But what I think it's ongoing, but I guess it won't take that long for them to have it public, is their prediction application. So in this part, you will be able to use genomic information and predict the design and the media that you can use to cultivate and isolate the specific organism of interest. So you would be able to go from the genome to the medium design and achieve, better and more successful cultivations. So the next step on the different way, let's say, to try to use omics guided cultivation approaches is to do a network guiding cultivation. So this case is a bit similar than the previous one, because again, there is a medium preparation step, but the whole idea is that sometimes it's difficult to isolate microorganisms on their own. We need to identify the potential interactions between the different microorganisms in order to cultivate them properly and more efficiently. So a way we can do that we can employ this tool. It's called MicroTag. It's also under development, but its a beta version is being tested currently. And it will be able to provide this information on the potential interactions between members of a community. So it will be easier for us to know which kind of microorganisms in our sample need to be considered as pairs, let's say, so we can cultivate them at once and have a media only for them, and so on and so forth. Of course, the tools can be used for many different ways and reasons, but this is one way they can be employed. So what these two approaches have in common, and what is like the golden standard in our age for studying, for every microbial ecology study out there, is the metagenomic sequencing. So the metagenomic sequencing, as you can see also in the picture, it starts from sample collection, then we do DNA extraction, we fragment our DNA and we sequence it. Either it can be an Illumina platform, it can be a Oxford Nanopore platform, whatever the technicalities. The notion is the same and the idea is the same. What we can do with the results afterwards, we can go through the assembly step. We can try to merge all the little pieces of the sequencing together and create what we call contigs. And then we can try to connect all the contigs and try to build what we say a genome assembly. We can go through the binning step where we can try to find, we compare our sequences and try to, we do this genome sequencing, and we can have like a genome codec coverage profile. Both methods result in taxa abundance profiles. They result also in functional abundance profiles. So we have an understanding of what is out there and what basically is their potential of doing. We don't know for sure. So one of the tools that we have developed in order to do that step of the analyzing of the shotgun metagenomic sequences is called MetaGoFlow. It was developed through a NEOS Clive project, and it basically builds on the Magnify pipeline, and it tries to shorten the processing way. So we designed it in having in mind the IMOBON data that I showed you in the beginning from the genomic observatories. So we had in mind that we had those data, and we were trying to identify who is actually in our samples, what microorganisms are there, in a fast and efficient way. So this is what we did. The workflow has several steps, and step 2 is, let's say, the one we were more interested in, which is the taxonomic inventory, and it's like a fast way to have the answer on what is out there. You can also complete the other steps, the assembly, the gene prediction, and the functional notation if you want. And in the end, you have a narrow crate build, which is a package, let's say, containing all your outputs and data provenance. So what has happened on your data until these outputs were produced. So yeah, if you want, you can take a look. This is the GitHub page of MetaGoFlow, and this is the docs documentation page. Of course, we built it having genomic observatories data in mind, but it can be used for any and every shotgun metagenomic sequencing dataset. So what we can do if we have, those sequenced data from our environments, then starts the fun part, and trying to identify, to have your questions and try to see, to do the science, the actual science, let's say. So one question people might answer is if all extreme environments are the same. If you have the concept of normal in mind, then all extreme might seem relevant and might seem more or less similar or identical. Yeah, so in order to do that, I compiled a list of data sets from different extreme habitats like hydrothermal vents, hot springs from a hypersaline lake, from a saltern, and from an acid mine drainage. Of course, this is by no means a complete list of everything that is out there, from not even from every data set or from every type of extreme environment, but that is also because data are not fair. So data are not findable, accessible, interoperable, and reusable. There are tons of sequence data out there at the moment because we have been focused on that part of research, doing short-term metagenomic sequencing for many, many years now. But the vast majority of this data are practically useless because they are not documented properly. They don't have metadata attached to them, so they are actually non-reusable, only maybe even by the researchers that actually produced them. So just, yeah, just to have in mind that the better you are, let's say, into having fair data and opening your research in the world, the better scientific results and conclusions might be formed, the better knowledge that we will be able to reach. So having, taken data from those sources, we can see, for example, that the highest number of species or for taxa at the lowest taxonomic level were found in the acid mine drainage sample. And the lowest was found in the hypersaline habitats. But what is striking is that there are no common species between the three habitats. Now, species is in quotation marks because we are talking about the lowest taxonomic level that the analysis reached. So it can be genus in some cases. You can, if you know this data, you know more or less what I'm saying. So yeah, we can see that there are no common species between those three habitats. But when we go to the higher level, when we go to phylum level, we see that this image is somehow changed. Now we find that there are indeed some phyla that are common between all the three habitats. Now, this is more or less something that we could see in every data set, regardless of it being from an extreme environment or not. We would see that there are similarities at the phylum level, but we would also see them in the species level or in the genus level. We wouldn't have seen so many differences. So yes, we have different results when we have taxonomies at a different level. The highest number of phyla in this case was found in the geothermal habitats, so in the hot springs and the hydrothermal vents. And we have shared phyla between all the three. Among them are Briarchaeota, Nitrospira, Firmicutes, Actinobacteria, and Protobacteria. So the image is slightly different when we go to the functional profiles and when we try to identify to see if there are similarities in the functional aspect. We see, for example, if you see in orange, we have orange or slightly brown. I don't know how it appears in your screens. We have the geothermal habitats, but we can see that they are not, for example, all of them clustered together. We can see that hot springs are in the bottom of the PCA plot, so they are clustering far from the other geothermal habitats. So even in the same extremity, we have differences. And we can even see that hydrothermal vents are clustered separately. So we can see the Jan Mayen one in the left part of the figure, and on the right part of the figure, as I see it, of course, we can see the axial mount hydrothermal vent. So we have different locations and different microbial communities. So although we saw before that when we have the taxonomy, we see that each habitat is more or less unique, we see that they are much closer from a functional perspective. So in this case, we see that they share several thousands of gene ontology terms. So they are closer functionally than they are taxonomically, if this makes sense, which is something to think about. So extreme environments may not be the same, but it also depends on how you look at it. So in the last part of my talk, I think I have a bit more time. And since we are dealing with climate change and global warming, just I thought about presenting some things on how extremophiles might relate to global warming. So what we know that temperature has been found to be a key factor affecting ecosystem carbon cycling. We know that increased temperature can change microbial physiological properties and decline the fraction of assimilated carbon that microbes can allocate to growth. So it has an effect on them. But we also know, and we also see that microbial communities in geothermal habitats that are already adapted to elevated temperature because this is where they actually thrive. And they could provide an assessment of how resilient and how tolerant such communities can be to changing environmental conditions. So in other words, if we study them, if we study geothermal habitats and we see their response to climate change, to increase temperature, increase gas emissions, we can perhaps try to understand how ecosystem function will be in the future, and we may even try to identify climate ecosystem feedbacks. We know, although from microcosm studies, that moderate temperature increase, so about 2 degrees Celsius, may not have a clear effect on microbial communities. But this is again, this is to be discussed. It depends on which microbial community was sampled in the beginning and was subjected to the experiment. How fast was the change induced? Were they able to adapt over time, or was it, very abrupt? So these are questions that need to be thought of, because climate change and global warming is not happening abruptly. It happens slowly every day. So it's something that we might have the chance to get adapted to, or not. We'll see. So according to IPCC, the Intergovernmental Panel on Climate Change, there are 4 possible scenarios on how much the increase in global mean surface temperature will be by the end of 2100. So in the, let's say, the less extreme scenario, the expected increase is up to 1.7 degrees of Celsius. And the more extreme scenario can be close to even 5, like 4.8. So if we want to assess, let's say, how this might affect our communities, we need to, we may need to use some specific tools that we can, towards this task. Some of the tools we can use, for example, are software that can give us the optimal growth temperature. They can predict, let's say, the growth temperature of our organisms, and hence we can have a community-wide growth temperature prediction. So some of these tools use taxonomy and are based on taxonomy, and this may be a bit tricky because when we do such studies and when we go on shotgun metagenomic sequencing, sometimes it's really hard to actually get identifications up to species level, because we might find many new species, many new genera, and so on and so forth. So in this case, it might be more appropriate to use tools for growth prediction that are based on the actual DNA sequences. Also, one thing that I have done for the purpose of this presentation is to go through the Tempura database, which is a compilation of growth temperatures of usual and rare prokaryotes, and try to see, and based on this data, how climate change might affect microorganisms. So based on this database and based on the compilation of genomes that they have there, I have plotted the average growth optimum temperature versus the genome GC content. So you could say that there is a correlation between the growth temperature and the GC content, but I don't think it's so clear. So this is why I haven't, drawn a regression line. If we try to plot on the graph the average surface temperature on Earth at the moment, which is 15 degrees, we can see that, okay, one would say that it wouldn't affect practically anyone, any microorganism. But then again, you would need to think that what is average doesn't mean that is true for the majority of habitats out there. We talk about extremes and we talk about environments that can be, their starting temperature can be either very high or either very low. So having that in mind, you need to, when you do your predictions and when you try to do your estimations for the future, you need to know, let's say, your starting point. But if we take a starting point, the average Earth temperature, and we try to see how it will become in the first scenario, you can see that I have drawn another line in the graph. In the second scenario, it can go very close by. The change in those two is not that different. And let's say in the final one, we can go up to 20. So let's say if we had a starting temperature of 30 degrees, or let's say that would be a normal temperature for a lot of environments on Earth. Then adding 5 to that, it would maybe create a big change. If we start from the average, which is quite low, then it seems like there will be no effect with global warming. But then again, it's climate change is a very it's a multifactorial thing that cannot be solely taken into account by temperature rise. So I think this is the last of my slides. Yeah. So if you want, you can, if you have questions that we cannot answer on time, you can always email me or find me on social media. So yeah, if you have any questions, happy to take them.
Very good. Thank you, Christina. Thanks a lot for a wonderful webinar. And thanks everyone for joining in. We actually have quite a few questions already in Q&A box and very interesting ones. So I'll start asking the question, but before we go there, I'll just like to tell everyone that this webinar is part of a series exploring microbial ecosystems, and there are several webinars coming up, so please explore our website, and I'll also send you the link where you can see other webinars and register for them. Okay, so we'll start with the first question. Can we use the extremophiles in planet exploration studies? I mean, can we inoculate the extremophiles using satellite technology in Mars or other planets?
That's a good question. You mean try to collect samples from Mars and try to cultivate them here on Earth, I suppose, not something happening in situ on Mars. That would be really interesting. That is something that there's a whole field of astrobiology and there's a whole field of exploration in that sense. And it would be really interesting to try and cultivate something from something with extraterrestrial origin. Yet it's also very, it can be also problematic to sample and bring it back to Earth, because there was a paper, I don't remember when, that was for the microbiome of the actual Mars rover before it was sent to space. So they actually went and sampled the Mars rover and sequenced it so that they would know that then when it came back eventually, if they would find whatever new they would find, if it was like from Mars origin or if it was like contamination from Earth. So in other words, it's difficult. I think the contamination aspect would be very difficult in that sense. And that we don't really know how life can evolve in other planets. We only have ideas. So yeah.
Good, thank you. Can extremophiles be superimposed by considering human microbiomes as one? And can the existing protocols that are used for environmental samples be used for humans?
Well, protocols can be quite similar. So whatever you can use for, let's say, extraction-wise from a sample in an extreme environment, you can use it also for human microbiome. I don't see a reason why not. But then again, okay, if you work on something, you can on a sample matrix, you know its peculiarities and you know more or less what you need to change. Yeah, I don't remember the first part of the question.
Yeah, so it was about like what you're doing for microbial study. Can it be used for human microbiomes, I think? So I think you answered this.
Yeah, but yeah, if you consider the biotechnological potential of extremophiles, then you know already that there's a lot of human exploitation and yeah, going on there. Yeah.
Okay, so next question is about climate change. Are acidophiles and or thermophiles now thriving in habitats where they weren't previously found? For example, in coastal environments or in certain geographical locations? Are there any examples for that?
You mean examples of climate change um, having made a shift to the communities already? I think that's the question. I don't know if anything related to that is actually published already. But if you think of climate change only as temperature rise, then it's really hard to uh, identify its effect on microorganisms as we speak. But if you think of climate change as increased heat waves or as increased rainfalls or flooding and so on and so forth, these extreme events that are happening in a faster rate, then yes, you can see changes in communities and microbiomes. Yeah.
Next question is about the analysis itself. Can you please expand a little on functional analysis through metagenomic sequencing using extracted DNA? Is it RNA that needs to be extracted for sequencing, or?
Yeah, it's ideally you could have both. You would have both, and ideally you would also have not only nucleic acid. You would go also try to go in the protein level, and yeah, you would need to compile information from multiple, let's say, fields. With RNA, what is the good thing with having RNA is that you have a direct link to the actual function. Now with DNA, we only have the functional potential. And bear in mind that with DNA, we don't really know, unless we treat the samples in a certain way, we don't really know if what we are amplifying and analyzing is dead or alive, meaning dormant or actually thriving. So we cannot, in some cases, we can make this separation. In some others, in the majority of cases, people neglect or they don't focus on that. So we assume that everything is alive, which is actually not true. So RNA gives you a better understanding of communities and their functions. But it can be also very, very hard to extract RNA from such environments. It can be even hard to have enough DNA for shotgun sequencing for some of these environments I mentioned, because they can be very limited in biomass. So yeah, so you cannot have, let's say, everything. It's not easy.
Okay, so next question is again about the analysis, and I would like to slightly interpret this. So one is about how can we use, I think, metagenomics data or Magnify to look for specific enzymes, especially when you are comparing, let's say, normal temperature and extreme environments. So can you identify specific enzymes which enable these microbiomes to survive in extreme conditions?
Yes, you can see different genes, functional genes being enriched or not. You can have such information from all the functional notation pipelines, so you can understand. You can see if there is a clear difference, especially if you compare it with, let's say, more normal sample, not extreme. The thing is that with gene annotation and functional annotation, there are many things that we don't know. So while we do have some of the genes annotated and we don't know what they're doing and what their function is, we have many more that we don't know, and we just have hypothetical protein as a result. So which is very vague, and it can be like a million different things. And there are tools specifically designed or that are trying to deal with this thing on how to annotate the unknowns. Yeah.
Is it easier to apply adaptive laboratory evolution to extremophiles in comparison to others? In other words, are they easier to adapt to new stress conditions?
That's a good question. I would say that it depends on the stress, because if they are normally, let's say, thriving under high temperature, so if their stress is high temperature and you try to apply something totally different, then they might not adjust as fast or or the way you want. It can be, though, that extremophiles are more ready to adapt in certain conditions and in certain changes than their normal counterparts. That it can be true, but it needs to be, let's say, tested in a laboratory scale, before we can conclude that.
Okay, next question was, have you designed a novel culture media for your samples?
I wish. That was my idea. That was one of the things I wanted to do for so long. What I have done so far is to do it only theoretically. So what DSMZ is proposing to do with the media dive and their prediction tool to get from the genome to the media, I have tried to do it in a smaller scale, manually, let's say. But I didn't go through with the final experiment. So I have used their platform. I have used MetaDive to search for recipes for media for certain tags of interest. That part I have done. Yeah.
Okay. In future, can tempura have a collection of also eukaryotes like fungal species that are also adapting to higher temperatures?
That's a good question. I think I'm not involved in that one, so we'll need to take that up to the actual people that design tempura. But it is very interesting. Fungi in general are quite interesting because they can be also very versatile, let's say, metabolically, and they have been kind of neglected in microbial ecology studies, let's say, more or less in general. I don't want fungi people to cancel me, but they have been so far. Well, while they can be really interesting, so yeah.
Okay, thank you. Next question is about the FAIR data. Are there any guidelines on how to make data more fair? For example, guidelines on what metadata needs to be included. Do scientific journals require that the data is fair upon publishing?
That's a good question. Journals generally don't require practically anything or require the least that they could. I have seen papers coming out from journals that they don't even require the accession numbers for the sequence data, which is insane in my opinion, if you ask me. So I would say the first step to make your data fair is to use when you try to submit it, to use one of the mix checklists, the minimum information for a sequence checklists that exist, and they are decided upon from the Genomic Standard Consortium. You can find them when you submit your data, your raw sequences, or in ENA, in NCBR, whichever repository you choose. I think that's the first step. If you want more information on what to do that, how to do that, there are some fair data courses. There was one organized like a month ago in the Flanders Marine Institute in Belgium. It was funded by EMBL-EBI. So if you Google it, you will be able to find a link to that. I'm not sure if the material is online available, but keep an eye on that one. Yeah.
Yeah, and I would like to add, I mean, from the training team, we also have published like article about how to make your data fair, and we also have some tutorial. I'll share those links when I send an email by tomorrow, so you will get the link to those. Okay. One question is, I'm working on microbes in soil of semi-arid and desert environment. Can I consider these as extreme environment?
Yeah, for sure. For sure, you can consider them as extreme. But yeah, then again, is let's say, what would that? It will be like an added value to your research, make it a bit more, let's say, an interesting subject. But yeah, definitely.
Okay, another one, will the extremophiles would act like normal microbes under protective environment like microplastic biofilms in water bodies, or they can change the growth of other microbes?
Well, under a protective environment, you mean like a normal environment, not so extreme like a normal environment, I guess. They wouldn't like it as much. So let's say if your optimal growth temperature is 60 degrees and somebody tries to force you to grow at 20 or 25, then probably you wouldn't even grow that much, or you would need some time to adapt to that, to adjust to that. Yeah, so I don't think they would be capable of growing and changing the growth of other microbes. At least let's give them some time to adapt.
Okay, thank you. Next question is, I'd like to interpret it that what are the main challenges in shotgun data analysis, and does it change if you use different platforms?
Yeah, that's a good question. Let's say the issues that you face when you try to analyze shotgun metagenomics data are most of the time the computational capacity that you have. So it's not very common for researchers and institutes to have, high performance computing clusters to analyze such data. I think that's the main thing, because you can find several different tools and pipelines to actually do the work, but it's computational power and time, which is also a very important thing, because if you have a big assembly to run, then it will take a lot of time to be completed. I would say that you can submit your data to ENA, promotion of EMBL-EBI. You can submit your data to ENA, and then you can request them to be analyzed by Magnify. This can happen if the data is private also, doesn't have to be public. So they can do the analysis, let's say, for you. It can take some time. If you want assembly, it can be less if you don't want the assembly part, but that's a good, let's say, opportunity if you don't have access to any high performance computing clusters. And it can change depending on the platform you use, because Illumina platforms produce fast-Q sequences. Oxford Nanopore has a different chemistry, a different way of sequencing. So it produces different raw reads, different type of raw reads. So there's a different tools are needed to analyze them.
Thank you. Next question is from a PhD student from Sao Paulo, Brazil, that I'm currently working on study of microorganisms of cave here. Do you also perform metabolomic studies? Could you suggest me free platforms to analyze results of LCMS or GCMS data?
I haven't worked on any metabolomic data. Although at some point I had found what you're asking. I had found platforms to analyze such data online. I don't remember them by heart, but again, not self-promotion, but I have a list of pipelines and tools and platforms in my website. So if there's a high chance that I have included them there. So yeah.
Yeah, and I'll be sharing the slides along with the recording, so you can get all the links from the slide.
Yeah.
Okay, very good. Okay, so great talk. Could you please point to any studies or examples of extremophiles potentially spreading antimicrobial resistance?
Yes, the paper that I cited about the caves and in the challenges slide, it's not. It's about the cave microbe that had been found to be in one of the deepest and oldest caves. It was found to be resistant in practically all antibiotics. So it hasn't escaped the lab yet, but it would be really problematic if such thing happened, because if you have a microbe randomly being among your normal communities and spreading the resistance to every single antibiotic that we use, it could be very devastating. Yeah, it's that study on the challenges slide.
Thank you. Another interesting question. Has there been any industrial usage of metabolites from extremophile microorganisms?
Oh, there are several. People have been trying to harvest extremophiles for several different reasons. They can, they have even found, let's say, from microorganisms living in permafrost or in glaciers. I don't remember which one was it. They can find enzymes to do many different things, even being used as detergents or even being used, to decompose strange things. So industrial application is something that I'm not particularly interested in, so I'm not following that part of literature very, very closely, but I know that harvesting is continuous for biotechnological applications for extremophiles.
Okay, thanks. So we still have just 3 minutes left, so and we have 2 more questions we can go through. Do really the network, core microbiota, and differentially abundant taxa analysis based on amplicon or MAG, the shotgun sequencing, really show what exactly happening in extreme environments? How can we confirm these analyses at wet lab level?
Yeah, that's a good question. Well, they can show in, yeah. We have to, unfortunately, this is our main, let's say, window to their world, to the microorganism world. This is like the main ways we have to study them. Of course, whatever we confirm, in the lab and whatever we have, whatever actual experiments we do in the lab are very, very powerful and useful. It's not that easy to work with extremophiles in the lab because they may require different alteration in the culturing conditions. For example, you cannot grow thermophiles in agar plates because high temperature will melt the agar and you need to find a way to replace it and so on and so forth. But there are always many experiments you can do in the lab at the moment. You can even grow them under in incubators with increasing temperature, with increasing, let's say, gas emissions. You can do whatever you want to do, depending on, of course, budget and equipment-wise. Yeah.
The other question is, slightly related, but from the other angle. What should be taken into account to improve sampling of extremophiles? And I would like to know your opinion regarding in the era of metagenomics. What do you think about isolation of microorganisms? Is it still important?
Of course, it's important. It's very important. It has been in my mind and in the way that I have perceived it. When we first started the high throughput sequencing, and this world opened up, people kind of neglected isolation, or they went a step back. And now we're trying to go a step forward and try to make up for the lost time. It's very powerful if you actually try to isolate the microorganism and try to have it grown in the lab. Imagine that it's the final, let's say, confirmation of your studies and your work. If you see the papers of Thai Setema group on Loki archaeota and the Asgard archaeota in general, they first started with metagenome assembled genomes. So they found them through metagenomics. And 20 years later, not 20 years later, but something grew in a bioreactor that was actually an Asgard archaeum that had been going for 20 or 30 years, I don't remember. So isolations are very, very important, even at this point, maybe even especially at this point that we are now.